It's great to be here. I'm honored to have this opportunity to tell you some basics of cryo-EM and also tell you what we are doing at Harvard Medical School. Thinking about method to determine high-resolution structure, there are essentially three methods right now. One is traditional X-ray quick crystallography. The other is MMR. And now is the new player of single-particle cryo-EM. So when people say cryo-EM, usually they only mean single-particle cryo-EM, which is the focus of today's talk. But equally important, electron cryotomography is also under revolution, and improving the resolution and particularly useful to look at, for example, the cellular structure, and also the very large complex, which is almost impossible to find identical copies. But we'll talk about single particle today. So today my talk will be three parts. One is talk about the basic practice, exactly how we do single particle cryo-EM, and what's the difficulty, and where we can further improve it. Then I will talk about two factors which really bring in the cryo-EM revolution, and I will use the project I was involved, like you mentioned, TRPV1 as an example, to show how a structural study of ion channel can benefit from that. And then I will go through three projects we are actively working on right now. I think many of you probably familiar with light microscope. So in a way, electron microscope is also optical. So for light microscope, the light source coming up from here goes through condenser lens, pass through specimen, and goes through objective lens and ocular lens, and finally reach your eye, so you can see it. And electron microscope is very similar if you look at the way from upside down. So electron source coming from here and is condensed by the electromagnetic lens, pass through the sample and goes through objective lens, projector lens, and finally the image can form as in the phosphor plate, which you can directly observe with eyes. If you lift the screen and let the electron pass, then you can have a detector, or simply called camera, and down there to record your image, which will allow you to do a lot of processing afterward. Now, for single particle cryo-EM, the sample has been prepared in a specific way. So biology sample, and in many of our study, is mainly purified protein, has to be embedded in a thin layer of Vitrobot eyes. The way to prepare this is put a small drop of protein on the surface of a holy EM grid, and you blot most of the sample out away by a filter paper. Then what's left on the grid is a very thin layer of liquid film. So this can be plunge freeze into liquid ethane cooled by liquid nitrogen at cryo temperature. So next, I'll use a video to show you exactly how this is done, and hopefully will give you more ideas about the size and of everything. So it's actually a thesis movie from Gabe Lander, currently at UCSD. So here is the sample. Here indicate the molecule is tumbling inside the solution. If we freeze this, there will be too thick for the electron to pass. So we put on this small grid. Diameter is only 3 millimeter. You can put several microliter of sample on it. And we have a semi-automatic blotting machine. And in this chamber, it can control the humidity. And you have two blotters with filter paper on it. And then you basically slam the two pieces of filter paper on your grids, and that absorbs away most of the liquid, and then the grids will plunge freeze into liquid ethane. the freezing is very fast, so crystal cannot form. You only get vitreous ice. So as a thin layer. So this thin layer of ice with the protein inside will be loaded into the microscope. And then you can expose it with electron beam. And then in the electron detector, you will have this so-called transmission EM image. Because biology sample is very sensitive to radiation damage, since it's only contain the atoms like nitrogen, carbon, oxygen, it's unlike heavy metals, so it cannot survive a lot of radiation. So that's why there's only very limited dose you can put on the sample before you completely destroy the protein. And then the result is the signal-noise ratio of the image will be very low. For processing, we have to pick out the particles. So here I circle several of them and extract them and form a particle stack. So each of these particles represent one single protein adopting certain orientation in ice. And this orientation has to be determined computationally. And when that is done, so all of them can be back project and average in 3D to form the original 3D object. With sufficient particle, you can suppress the noise, increase the signal, and achieve high resolution. So if you are familiar with crystal, in a way you can think of this is a crystallization in silico. Because you don't require the crystal to make them order, you actually use computation way to calculate its angle. I will emphasize that a low signal noise ratio of the cryo-EM image is the key to understand the whole technique. Why is that? First, that fundamentally determines that we have to care about protein homogeneity, because we have to average thousands, sometimes millions of particles to get the final structure. And the particle has to represent the same conformation, not only the same protein. They have to adopt the same conformation to be averaged and form the final structure. And computation algorithm is very important because every particle is kind of fuzzy. So we need very good algorithm to accurately determine the angle. And even better, we would like this algorithm help us to sort out actually different conformation. Because very strictly speaking, there's no two proteins are completely identical. So they're always identical to certain resolution. And finally, the electron detector is very important because there's only so much signal can reach at the bottom of the microscope. We want to get most out of every single electron. So a good detector will help us see the small particle better, and also it can preserve the high resolution information better. And precisely in these two aspects is where comes the cryo-EM revolution. One is the probability-based 3D classification dramatically improve the computation algorithm in terms of sorting out the particle and also improve the final resolution. And also the invention of direct electron detector basically boosts the resolution for most of the structure to the level that you can see the side chain and build the model. So next, I will use TRPV1 as an example to show how these two aspects actually help improve the result. So that's the work when I was still a postdoc working with Yifan Cheng at UCSF. It's a collaboration with Hu Cao at David Julius' lab. Hu now have his own lab at University of Utah. So Hu purified this protein and struggled for a couple years. So very difficult to get the crystal structure. But it's a terribly important protein. So it's a receptor for capsaicin. Capsaicin is the active ingredient of chili pepper. It binds to this receptor and generate a signal, electron signal, to indicate, to kind of generate this burning hot sensation. And it's also an internal heat sensor. So that's what we got using cryo-EM back at 2011 or 2012. So we collect, so that's still the traditional CCD camera. So we collect about 45K particles and throw them all into one structure. And we get something kind of makes sense and kind of doesn't make sense. So we have this feature, we don't know what they are. And here's supposed to be the soluble anchoring domain, but doesn't seem to be the right shape. So we have a strong feeling that our particle probably contain a lot of junk. But at that time, there's no effective way to separate the good particle from the bad particle. So around that time, probability-based 3D classification come into play. And particularly, Dr. Charles Scherer at UK developed now really the dominant program in the field called RELION. So we were one of the first groups to actually try this program. So using this same data set, and we asked the program to give us 6 classes, and that's what we got. Although at this stage, all of them seems low resolution, but immediately we identify only this conformation is relevant. All others is not relevant. And this class only contains less than 23% of particle. And only use about 10K particle, we were able to achieve a resolution at about 8.8 angstrom. So there we can already see isolated transmembrane helix and also very nice shape of anchoring domain. So at that time, if without direct electron detector, that also will be a record in that year. But I think at that time, cryo-EM is really being blessed. So, so we have another revolution coming on is with the electron detector. So nowadays, there are three companies sells this type of detector. And I would like to mention that all these detectors actually are developed by collaboration of academia and industry. So particularly for Gatan, K2 and K3, so at that time it's K2. So it's developed by a collaboration of several groups. And the first camera being tested on biology sample is right at UCSF. So I was lucky enough, actually, the TRPV1 is the second data set ever collected on K2. So using that data set, we were able to achieve 3.4 angstrom resolution. So here you can directly see the side chain along the transmembrane helix. So here will be a zooming view of different area. So you can see it's a great improvement essentially from 15 angstrom to 8.8 angstrom and then now to 3.4 angstrom. So we enter the regime that you can directly build a model and then explain a lot of biology. So after we solved the first structure and essentially the whole purification and structure determination pipeline is well established, we were able to quickly generate a set of additional structure using different compound and toxin. So the two are published one is a combination of 2 toxins to generate a complete open conformation for TRPV1, and the other is the capsaicin to generate a partial open conformation. So by lining up all these structures, that actually give us really the structural basis of the activation mechanism of this particular channel. So since then, we essentially observe, there are many words for this, explosion, avalanche of structure, or whatever. So here is the bar graph. So the total deposit cryo-EM structure after 2013 has been significantly increased. And if we focus on the structure which are better than 4 angstrom, which is at the level you can build model, is even more significant. Essentially, before 2013, you only have that level of structure for very large objects. And some of you probably know by today, there are already structure for every subfamily of chip channel. And the field is really going very fast.
I have a question.
Yes.
So for the membrane proteins, do we have to remove lipids?
You mean completely remove the lipid or? No, I don't think so. And you probably would even prefer to see what's actually stably bound on the protein. That would,
yeah. You have to worry about the lipids here with the imaging, and just you are picking one image for, for protein at a time. The presence of lipids with that.
So if the binding of the lipid is not going to dramatically alter the structure, it's probably not an issue. And I think I'm not aware there was one example of that. If, in fact, the thing is on the opposite, many people are super excited to see a lipid density when they after they solve the structure.
I think your question is whether your specimen for your protein actually was embedded in lipid.
No. So for the CHIP-V1 is in the amphipole. So it's some type of detergent, but it's bind very tight.
But couldn't be seen because it's disorganized.
Yes. When you filter to low resolution, you kind of can see the weak density surrounding it. Yeah. 2007, so Nobel Prize was awarded to 3 scientists, which is have a significant contribution to cryo-EM of high-resolution structure determination of biomolecule in solution. Essentially, these several words is actually defined. That's the technique of single particle. So I started my lab at 2014. So here's where the building I am in. So it's called CLG-MUD. So here's the quad of HMS. If you have been there, so you probably can recognize it. So we now have actually 2 cryo-EM facilities in that particular building. So here is the old facility which I have been using, and I'm going to show you a set of structure, and most of the structure, the data is generated by this Polaris 300 kV, which now is already very old, but it still gives us very good data. And just in this year, together with several other institutes, now there's a new facility just started. So it hosts 2 Titan Cruise and 1 Telos Arctica, and all of them have the most updated K3 camera. So we are all very excited about this. Now I would like to tell you what we actually do at HMS. So my lab is focused on using single particle cryo-EM to study DNA protein complex and also membrane proteins. So through collaboration, we generate high-resolution structure for RAC12 complex and also CRISPR complex. And collaborating with other groups, we generate structure of the ER-rich transport channel and ATP synthase, and also mitochondria uniporter. For the rest of time, I will focus on our own research, and which is mainly focused on a specific group of transporter. And this group of transporter is called ATP-binding cassette, or ABC transporter. So this group of membrane protein is probably the 2nd largest membrane protein family, just smaller than GPCR. So you can find this member in all kingdom of life. This protein always contains two parts. One is transmembrane domain, one is nucleotide domain, nucleotide binding domain. So nucleotide binding domain will sandwich two ATP molecule at the interface, and then induce tight dimerization of NBD, and then change the conformation of transmembrane domain. And transmembrane domain is responsible for binding and translocate specific substrate. Different APC transporter can transport different substrate. It has certain range of specificity. It can transport iron, amino acid, sugar, lipid, and even small proteins. Because it's very small and also it's undergo a large conformational change, so it's very flexible in a sense. So it has been very difficult to study by structural approach. So in terms of the size, it's about 130 kD for the two proteins I'm going to mention. And it has 12 transmembrane helix. So just give you kind of a comparison. So for NAV channel or sodium channel, so it will contain 24 transmembrane helix. So that gives you more signal from the membrane. And also the size is over 200 kD and usually up to 250 kD. I'm going to tell you, briefly tell you our result from two bacterial LPS transporter and also one human multi-drug transporter. So here is the schematic of LPS, or lipopolysaccharide. And if you are not familiar with this name, you probably know the name of endotoxin. So it's a lipid at the gram-negative bacteria outer surface, which is can also is a main inducer of immune response during the bacterial infection. So this lipid is actually very large and also strange. It contains 6 tails. So 6 tails attached to 2 glucosamine, and each of them attached to 1 phosphate. And this lipid is decorated by a long chain of oligosaccharide, which can be further divided into 3 groups: inner core sugar, outer core sugar, and repetitive O antigen. So at the surface of gram-negative bacteria, it's actually the major lipid component. Because of this charge and hydrophilic property of LPS on the surface, it's generally a very different property compared with regular phospholipid, which is generally hydrophobic. So because of these two very different properties, it's come together and very effectively exclude most of the antibiotics from entering the cell. So it's very interesting to understand the LPS transport biosynthesis pathway and as a target to develop novel antibiotics. So let's look at the steps of LPS transport. The nascent LPS is synthesized at the cytoplasmic interface of inner membrane, where the energy and building blocks are available. When they are formed, the first thing they need to do is flip to the other side of the inner membrane and be mature by addition of O antigen. And then it needs to transport across the periplasm and reach the outer surface. So it's a quite remarkable transport process for a gigantic lipid to pass through 2 membranes and 1 aqueous periplasm. So let's look at the first protein which is responsible for the flipping. So MsBA is a ABC transporter. It contains transmembrane domain, nucleotide binding domain. Nucleotide binding domain bind and hydrolyze ATP, induce conformation change, and then flip the lipid. So that's a general process. But exactly how that happens is unknown. Because when you think about this process very carefully, there are actually a lot of interesting questions. So the structure has been solved more than a decade ago. So the structure in the detergent basically shows there are a range of conformation the protein can adopt. Depends on whether it bind nucleotide or not. However, the substrate, LPS, is actually not resolved. So based on this set of protein structure, a model has been proposed that before the nucleotide binding, it adopts the inward-facing conformation to capture the substrate. And after the binding of nucleotide, it converts to open to the other side of the membrane and release the substrate. And this model is called alternating SS model. But because the LPS is not being resolved, so we don't really understand how the protein accommodates such a large size of substrate. And the lipid have very strong hydrophilic and hydrophobic portion. Exactly how one protein can handle a different property of the same substrate is unclear. And a big mystery is how this lipid can undergo 180-degree rotation. Does it do this completely inside the protein or have part of the substrate stick outside the protein to accomplish this is unknown. And finally, we really want to get a complete picture of how three aspects actually combine onto the one protein machine. ATP hydrolyzing cycle, conformation change of the protein, and the substrate translocation. So now this kind of back to your lipid question. So we are a big believer of lipid membrane, and we believe membrane have a fundamental regulatory role of any membrane protein. So the method we prefer is actually combining lipid nanodisc and single particle cryo-EM to study membrane protein in a small patch of membrane. So for people who are actually not familiar with this technique, so it started by solubilizing your target protein, the lipid you want to incorporate, and also a membrane scaffold protein, which is derived from apolipoprotein. You mix all this in detergent, then you remove the detergent either by adding biobeads or doing dialysis. And then all these components will self-assemble into a nanoparticle. So your target protein will be surrounded by a small batch of membrane, and then that will be further surrounded by the membrane scaffold protein. So this nanoparticle is amenable for single particle cryo-EM. So using this technique, first we reconstitute the MsbA and measure its activity to hydrolyze ATP. So what we found is with a set of different components to form the nanodisc, they always show much higher ATPase activity compared with the protein purified in detergent. So then we focus on this particular sample and solve a structure at an overall resolution at 4.2 angstrom. So we were very excited to see that actually there is a density inside representing the LPS substrate. So we were able to put into the important chemical group based on the density. And the connection of inner core to one of the glucosamine ring allow us to separate one phosphate from the 4' phosphate. And because of the flexibility, only a portion of the lipid acyl chain was solved. So a very interesting finding directly from our structure is that the LPS originated from here, and in our structure is actually already trapped deep inside. So the lipid acyl chain of the substrate is actually already reached the target leaflet. The only thing it hasn't done is the 180-degree rotation. So how does the LPS actually reach this point? So in our structure, the best resolution portion is actually the part which is surrounding the glucosamine and the attached phosphate. If we take this part, rotate 90 degrees and look from top down, and we are able to see that surrounding these two phosphate is actually a ring of hydrophilic side chain, and almost all of them are positive charge. So essentially, the protein generates a positive charge trap to elevate the LPS toward the target leaflet. The other interesting thing is 1-phosphate is actually almost exclusively interacting with the hydrophilic side chain only from one transmembrane domain. And that immediately indicates that the stable binding of LPS actually hold the two transmembrane domain in position and probably stabilize the whole conformation. We also solve another structure by using ATP vanadate to lock the two MBD at a very tight dimerization conformation. So in this conformation, the transmembrane domain also undergo a large conformation transition. So LPS is gone. Presumably has been flipped out. So that indicates that this conformation represents a conformation which is after LPS flipping. And there essentially no empty space in the cavity. So with these two structures and a set of function study, we come up with a model to describe the whole process of LPS flipping. So I'll show you a movie to illustrate that. So that's an inner membrane, and MsbA without binding of LPS supposed to be very flexible, and LPS can be trapped inside and stabilize the whole protein conformation. So that's the state we solve the structure, and this conformation is also can promote the binding of ATP. And the ATP binding will induce the tight dimerization of MBD and also induce the opening of TMD. One detail here is originally this lipid acyl chain is facing kind of pointing up. So when the transmembrane domain is opened up and directly exposed to the aqueous phase, so that generates a very unfavorable environment for the lipid acyl chain. So we suspect this type of the rotation at this step is probably happens spontaneously. So after LPS is released, transmembrane domain will collapse and forming a helix bundle. So that would be the structure we solve here. So after we published this paper, we further improved the resolution of this conformation. So now we can reach about 3.4 angstrom. So we can actually directly see all the side chain of the helix. And after this, the ATP hydrolyzed and released, and the protein will get reset into the original conformation and get ready for the next cycle. Yes?
Did you try and solve a structure without LPS?
That's a very good question. It's actually very hard to get a protein without LPS bound in it, because when we purify, most of them actually stably bound with LPS. And we are trying a couple of approach trying to strip off the LPS and see what's the conformation it is without LPS inside.
It's produced in E. coli or
yes.
Mainly? Yeah.
Mm-hmm. All right, so that's the MsbA story. Now we go forward in the pathway. So for this cross-periplasm transport is mediated by a set of 7 proteins called LptA to G. And in the membrane, there are three proteins, LptF, LptG, and 2 copies of LptB, actually form a minimum ABC transporter. So this complex can bind and hydrolyze ATP and extract the LPS from the membrane and deliver it to the protein acceptor domain. BFG complex is stably interact with a single membrane, single transmembrane protein called LptC. LptC have a large soluble domain interacting with one of the protein of F and G, and further interact with a soluble protein in periplasm called A, and then further interact with outer membrane complex called D and E complex. So so now the study or the evidence suggests a model that all these proteins actually form physically interacting bridge to transport LPS from this membrane all the way to the outer surface. So for our study, we focus on this minimum ABC transporter and also transporter associated with C. So first, we'll look at the BFG. Essentially, we take very similar approach as MsbA. We overexpress and purify these two complexes from E. coli and reconstitute them into nanodisc. So running on the gel, you can see the MsP1 protein. And first, we also look at the ATPase activity. So very interestingly, we have two findings. One is this complex in nanodisc is much more active than they are in detergent. Second is when we have the full complex with C, the activity is actually much lower. So that can only probably indicate two possibilities. One is this high activity is somewhat not relevant because we take out one protein. Second is maybe that's the true activity, but in the resting state with LptC, it's a self-inhibited state. To think about that more, we exercise ourselves with some number, but that just suggestive is not like the concrete evidence. So previous studies suggest that about more than 1 million LPS molecules need to be inserted into the cell surface during one generation. And that means we need to have 70K LPS inserted per minute. And from ribosome footprint assay, it's estimated every cell will generate 200 new copies of LPT complex in every cell. So that translates into about 350 LPS transport per minute per LPT complex. So that corresponds quite well with this number, which is 250 molar phosphate hydrolyzed per minute per molar complex. So that means if we hydrolyze one phosphate to transport one LPS, this number roughly makes sense. So we solved the structure of B2FG first, and we got a structure at about 4.0 angstrom, and that's have a sufficient resolution to resolve almost every single side chain in the transmembrane domain. And excitingly, we also resolve LPS inside. This density indicates the full inner core, phosphorylated glucosamine, and also 6 lipid acyl chain. I will emphasize that so this acyl chain is much more complete compared with those in the MsbA, and we can even see clearly the secondary lipid chain connection. So with all this, we were actually able to directly assign all 6 lipid acyl chain. So more detail. Here we look at the inner cavity of the protein complex and color the hydrophobic surface as orange and hydrophilic as blue. So you can see that the 6 lipid acyl chain is actually fit into a very tightly fit into a very small cavity. And if we look at the detail, so here's the lipid acyl chain. You can see how it have this very close contact with the side chain. So like this phenylalanine, and also the methionine and tyrosine here. So while we are working on this protein, actually there are two groups solve two crystal structure in detergent. Our structure is pretty much identical to one of them. And in this particular structure, the author claimed that they see some strong axial density in the cavity. They suspect those are LPS, but since it's very weak, they cannot do modeling. So compare this result and our cryo-EM structure, we think what happened is when the protein is purified, in their group or in our group, they always have LPS bound inside to start with. But during the prolonged crystallization process, LPS probably gradually dissociate. So the next step, we would like to look at the structure of BFGC. So taking a similar strategy, we solve a structure at overall resolution at 4.2 angstrom. And particularly, we have good resolution of the transmembrane helix. So we were able to build all the model. And also, we identify an actual helix, which is colored in red here. So it's a total surprise that this actual helix is actually positioned in between two transmembrane domains. And some detail about the interaction is this helix have quite extensive interaction with the TM5 from LptF. So all this interaction is through small hydrophobic residues like leucine, isoleucine. And also on this side, we also identify a lipid density, which we model as a POPG. So it seems like through all this interaction, the helix from LptC is actually have very strong hydrophobic interaction with LptF, and also have a weak interaction with LptG. So this surprising positioning immediately implicated one potential mechanism of the observed inhibition of ATPase activity in our ATPase assay. So we all know that when ATP bind here, it needs to induce conformational change of both MBD and also transmembrane domain. So this helix here is probably restricting the conformation change, so then that limit the ATPase activity. To study that directly, we started from this sample, and we add ATP in that in the presence of vanadate. So vanadate is a compound that after ATP hydrolyzed to ADP to phosphate, and gamma phosphate release, vanadate will comes in and form a stable ADP vanadate complex and then trap inside the active site, so that you would be able to study the structure when NBD is still tightly dimerized together. So we solved the cryo-EM structure of protein in that state. So you can see now the two NBD is tightly dimerized, and the transmembrane helix also kind of packed together. Interestingly, we don't see the C helix at all in this structure, although the elongated structure from the soluble domain would prove that this protein is there. But the helix becomes flexible in this conformation and cannot be seen. So here's a morph from nucleotide-free conformation to vanadate-trapped conformation. So the NBD conformation change will push the two transmembrane domains slide on each other and essentially collapse onto the inner cavity. So we suspect this will be the mechanism for the protein to squeeze out the bound LPS, although we are still in the process to directly visualize that step. So here's our working model. In the resting state, without the LPS binding, the two transmembrane domain is only loosely interacting with each other. And that allows the LPS to come in through the interface between C helix and the LptG. LPS bound inside will induce the conformation change of two transmembrane domain and make all those residue come closer to each other and forming a tighter organization. And this will promote the binding of ATP at the interface. So during this process, the C helix has to be removed or become less ordered at the interface. So that allowed the ATP binding conformation change to occur, which will squeeze out the LPS into the periplasmic domain. And then the ATP will hydrolyze and get ready for next cycle. And it's very likely during the active cycle, the C helix probably never get a chance to go back and stably bind again at the interface. So with additional cycle of translocation, there will be more and more LPS get squeezed into the periplasmic domain and then actually pushing the LPS into the periplasm. So any question for this part? Okay. So that will be my final part of the talk to tell you a little bit about our current study on human multi-drug transporter. So in the cancer therapy, so there is a phenotype called multi-drug resistant. So usually when a patient takes one drug for at the beginning, they're effective, and gradually it loses the efficacy. So some of that reason is because the cancer cell have overexpressed molecular pump or transporter, which can actively pump out the drug which inside the cell. So then the effective concentration of the drug inside the cell is very low. And so we would like to understand how this one transporter can transport so many different drugs. So here are just some example of the drug which can transport by PGP. And without the help of structure, people have struggled decades to generate 3 generations of inhibitor of PGP. But only until very recently, we have some structural information how this inhibitor actually bind and block the activity of PGP. So here I will give you one example of zorsocrida. So we saw one structure of zorsocrida bound to PGP at 3.8 angstrom resolution. So we didn't polish the map, so you can see a lot of bump here. But essentially, here shows we can see many side chains on the transmembrane helix. Inside, we were able to directly visualize the 2 molecules of zorsocrida actually bind onto each other. The shape is actually allow us to have an accurate docking of the molecule. So I actually put this chemical structure at relatively similar orientation of this molecule. The binding of this molecule actually induced large conformational change of the transmembrane helix, particularly a very large kink of TM4 helix and TM10 helix. These 2 helix comes into the middle and to the bottom of the drug and actually occlude the inhibitor from the cytosol. And surrounding this compound, there are many hydrophobic side chains, and most of them is actually aromatic. So we are really at the beginning of getting a set of structure of, yes?
Was it known previously that 2 copies of the compound are found?
No, the stoichiometry is unknown. Yeah. So it's a new finding.
And it's definitely 2 copies that you're seeing in the structure.
Yes. And also, that's not just our group, and actually another group also work on this and get very similar structure. So we are at the beginning to actually try to solve a set of structure of PGP bound with a set of inhibitor, also a set of substrate, to help us understand the structural basis of substrate and drug recognition, and also help us to understand why some of the compound function as inhibitor but not substrate, and why some actually do it the other way around. So hopefully, I have shown you that what we can do with single particle cryo-EM. So we were able to determine high resolution structure without crystal, and actually without perfect sample. And because of the power of 3D classification, we were able to characterize multiple conformation of dynamic proteins. And we can do structural analysis now in relatively native context. For example, membrane protein in lipid nanodisc. And we were able to directly visualize the compounds such as lipid and drugs with the potential to help the drug discovery. And finally, it's the same slides. So previous postdoc Wei Mi is generate the MsbA structure. He's now a PI at Yale. Student Francois is carry on the study of MsbA. LPT structure is solved by Yan Yan and Ben in the lab. And Min Luo is a postdoc working on PGP. And we were lucky to have a lot of EM and computation support and the funding. And thank you for your attention. I'm happy to take questions.